Hello, welcome back, and welcome to our afternoon session. Joris, our host, is going to say a few words for the opening, and then I will give you some further instruction about these afternoon activities. Thank you, Joris.
Sorry. Thank you, Laura. So for those who joined from overseas, welcome to this 3 hours implementation session on veterinary vaccines, batch release testing. Sorry. So this morning we had a full morning with interventions from regulators from all over the place, all over the world, and from industry. And I will be brief. So the most important points, in my opinion, is that all those we heard this morning from EDQM through USDA, through our Asian colleagues, all insist on the need for collaboration. That's extremely important. Secondly, we have seen in the, among others in the EDQM presentation, a fantastic overview on the 3 R's, which is already happening and which happened already before VAC2VAC and which is continuing. We have seen the same from the USDA with an approach a little bit different, but we understand that in the United States, a lot of things are possible on condition they are pragmatic, science-based, and proof of with results, with data. And very interestingly, from our Asian colleagues, we really welcome and are very happy to see the openness to the non-animal batch release testing and the 3 R's. From there, we can say if we exchange and talk to each other much more to understand the needs, because I insist always a collaboration is only possible if each party is capable to listen, not only to hear, but also to listen and to understand, and then try to exchange and understand each other really, and then you always come to a consensus. So I'm quite pleased to see the advances that have been made, and a little bit stressed to see what still has to be done, and a lot has to be done. But with a group like this one, and as I said, someone said I have a dream. So the dream that I have and we have in this 3 R's is that we on a global level can come to this consensus of harmonization, which will be to the benefit. We should not forget the 3 essential points on this. So animal welfare, of course, but equally reliability of the tests, repeatability of the tests, the consistency approach in the production of vaccines, and the control methods, which are not only more reliable, but also shorter, cost-effective, and finally, whether it's an animal or human vaccines, a better access to vaccines. So that's a short overview, the big lines. I stop here. Thank you.
Thank you, Joris. The objective of this afternoon session is really to hear a little bit from all the participants and to understand a little bit better what are their views on the complexity that still exists, and try to discuss together about the opportunities that we might have to overcome those barriers from both a technical, scientific, and regulatory aspect. And the way we would like to invite you all to participate is not only using the chat in Zoom, but also to, of course, speak directly. But to facilitate the discussion, we are going to use Slido, which is an online pooling website, and the way it works is that you will receive a link through the Zoom chat, and a QR code is going to be displayed. You just follow the link, and you will be invited to answer to some of the questions that we are going to pose to you. The idea of answering this question is also to make sure that we hear from you and we understand your perspective. There are two sessions this afternoon. As I said, one is dedicated to a discussion that is more focused on the technical and scientific challenges that are still to be faced, and the second one is going to be on the regulatory aspects. So with this, I just want to introduce the two moderators that are going to help us to discuss about the technical and scientific aspects of the challenges that are still in place. We have Paul Stickins from MHRA, NIBSC UK, and Botond Siklodi from Ceva. They are actually helping us with the discussion this afternoon. So I just invited you to open the link so we can start directly with the question. Paul and Botond, the floor is yours.
Thank you, Laura. So we both, Paul and myself, will be the moderator of this session. So it means that we don't want to speak too much. So instead we are encouraging everybody in this room and also who join online to participate in this session, actively participate. And the first question is already here on the screen. Obviously, this question can be answered by those who are actively involved in method development, either in the technical or regulatory phase, but answers are welcome from everyone. So please start using the Slido tool. And then the answers will be summarized and we'll see the overall results. So you have to make in order what is the most important, what is the biggest technical barrier or the least barrier. So the biggest technical or scientific barrier would be given number 1, and the last scientific barrier is number 4. 1, 2, 3, 4, 5. Sorry. Okay. Maybe we can limit the time of the answer, so 2 more minutes.
Almost all week.
Yeah, so we have more than 50% answers, and here you can see if you go back to Zoom, you can see the summarized results. You want to continue?
Yeah, well, it looks pretty even, certainly with the 4 specific options that were available, which I suppose is not that surprising because all of these are a challenge to some extent, and people will view them slightly differently in terms of their own experience as to what has been the biggest challenge. But just on the first one, because we have heard a little bit about this today on the need to demonstrate concordance between the in vivo and the in vitro test, and we've heard why that might not be scientifically justified. Catherine touched on it in her presentation. So I wonder, Catherine, if you'd mind elaborating a little bit more on the 5214 view on this. Thank you.
Thank you, Paul. Yeah, so as you know, as Paul said, we've talked about this a little bit already, and the idea behind the chapter is really to emphasize that when substituting in vivo methods for in vitro, the traditional idea that you need to have concordance or correlation is what we used to say between the in vivo and the in vitro result is often not possible. Sometimes it is, okay, sometimes it is, but often it's not. And in most cases, it's more related to the existing in vivo test than the quality of the in vitro test. As I said earlier, the traditional tests are legacy tests that were developed a long time ago, pre-modern validation science, let's call it. And very often things like the precision, the reproducibility, the limits of detection, quantification aren't known. You can maybe try and figure it out from your historic data, but it wasn't done upfront. In addition to the fact that the tests are often very variable. Rabies is the perfect example. The test in the pharmacopoeia for veterinary vaccines is a limit. You need to be within 25 to 400 confidence interval for having a valid test. So how can you compare a very accurate test where you might have a nice slope to a test where you have such variability? It's almost impossible to get a correlation or a direct correlation. So this chapter highlights those elements and explains why it is difficult or sometimes impossible to have this, and why it may not be necessary. It provides examples of alternative approaches for the substitution of the methods. It highlights that one way to get towards alternative approaches is multi-collaborative centers, and we've talked about those, but that's not a prerequisite, and that you can, as we've heard, independently in a manufacturer-specific situation, do this exercise and take it to the authorities and get it approved. So, for example, there's an example for potency tests and what types of things you would need to look at in order to make this exercise for potency tests. You have to understand the key quality attributes of your product and show that the batches that are produced afterwards are equivalent to batches that are shown to be safe and efficacious, either through the original clinical studies or trials, or in comparison to the batches that you've been using and have shown to be adequate for the last 20 years. And this is mostly important for the very long history vaccines. You should have an assay that looks at the antigen content, but also the functionality. And you should understand what you're measuring. You need to measure something that's relevant. If you measure an antigen assay that measures an impurity that's always there consistently, that's not going to be helpful. Or if it's an epitope that is of little relevance to the actual function, also not so helpful, unless you have other tests, as Kat said earlier, that can do the job in a different way. So this is another point. You may need multiple assays to get to the same answer. And that adds, in addition, the difficulty of having any sort of correlation, because you can't compare two tests to one test. So there were the examples with respect to that. There's examples with respect to safety tests, and they need to be specific and at least as sensitive as the existing method, but the way that that's prepared can take different forms. And in most cases, where an in vivo assay is to be replaced, there'll be data available already on the sensitivity of the existing model that you can look back to try and give you an idea of what to do. Sometimes the challenge there is if there's no reference material, so you don't know what you're comparing exactly, because a certain number of nanograms of toxin A and toxin B may not be the same activity, so that can lead to challenges, but that's something that has to be addressed during the exercise. There's the molecular consistency approach. Deep sequencing is mentioned. This is another newer approach that is given as an example in the chapter. And there you really need an in-depth knowledge of the molecular markers that are responsible for example, for the live attenuated vaccines, for what's responsible for the attenuation. And this is an ongoing exercise with polio vaccines. I think you're probably following that, many of you, as a good example for ways forward. And finally, there's also the detection of the extraneous agents by novel molecular methods is given as an example. So there's a lot of meat there for reflection, and I've heard already today 2 examples where this has inspired other documents and guidelines, and I think that's a good way forward. If we're all on the same page, then it's easier to move forward together. Thanks.
Yeah, it is. Thanks very much, Catherine. I can see why people have ranked this number 1, because if the expectation is there that you have to do this, that's a real barrier. But on the other hand, it becomes completely removed where you have the understanding within the authority that it's not scientifically relevant to do this. So it's a really interesting one for discussion.
And this is a good example when regulators come to help, because technically, scientifically, it's a difficult situation, it's a difficult task, but now regulators recognized it, and they allow industry to skip this step.
Yeah,
when it's justified.
Just 2 quick follow-ups, Catherine. Am I right in thinking that there's a plan to update the chapter? And 2, is it a chapter that's openly available?
I'm not sure about the plans to update. I know you're in group 15 and this is their baby, so you may have more up-to-date knowledge than I do. But let's say all pharmacopoeia chapters are open to revision if there's a justified revision. It has to go through the group. There's a proposal made to the European Pharmacopoeia Commission who adopts the work program, and then it can be worked on. So if there was a need, this would be possible. But I can't say the actual status right now. I think there's a lot of other work ongoing in the group that, and this may not be a priority at the moment, but I don't know. I can't say. And is it open access? It's part of the pharmacopoeia text, so it's not open access. No, it's part of the European Pharmacopoeia.
Okay, thank you. So I think we can move forward, and the next 3. Oh, sorry.
I'm sorry. I've just a point. I'm thinking about the others because I think it's very interesting. My position is that we cannot, most of the time, have a concordance of in vivo, in vitro tests, so forget it. The idea would be more have a good correlation of protection by the in vitro method, or a good consistency approach by good quality attribute, which is also very difficult to define. So that's my point of view, but this is the others.
Okay, thank you. Also, if you online have any more comments, then please indicate it. If not, then we can move forward. And the next 3 answers was given almost equally ranked. So it's adjuvant, and or maybe we can go to the second question if I see the screen.
I think we still have a couple of people who wanted to comment, and then Catherine as well.
Online?
Marianne Kaashoek from MSD. I recently had some experience in exchanging in vivo test by in vitro test for a couple of our products, and both products with adjuvants and without adjuvants. Adjuvants are really trouble for some of the antigens, and but as long as you keep looking, you always find a method to circumvent this. At least that is our fortunate experience. And about the correlation between in vivo and in vitro, that is an expectation by many authorities within EU, but also outside EU. And I think for all the in vitro tests, you have to show the dose response relation. So when you add less antigen into the product, then you really see that in your response in the in vitro test. These data are not often available for the in vivo test because it's very old and you try not to subformulate, but then at least you can show that your in vitro test is as suitable as the in vivo, and that gives you the reason to exchange that test and to continue with the in vitro successfully.
Yeah, no, thank you. Very good points. Just on the adjuvant question, are you referring to aluminium or non-aluminium?
What I know of is a product with aluminium and oil.
Catherine?
Yeah, I've sort of lost the train of my thought, but I'll see if I can pull it back. I mean, I've been thinking, and this is more relevant for new products, maybe, but just a philosophical question. If you were to drop down right now, you've never made a vaccine before, you've never, didn't have the history behind you, and somebody said, okay, make the vaccine, tell us how you're going to show if it's a good quality vaccine and if it does what it's going to do. Your first instinct would probably not be to go to set up an animal test, would it? It would be to really characterize the vaccine in a careful way and look to an in vitro approach. Or maybe I'm naive, and that I know for development of the vaccines, this is still an important point. But for the regular batch release, is that something that if you came now blind to the history, is that where you would go?
Well, I think recent history gives us the answer. You know, lots of examples of newly developed vaccines that have no in vivo element to their routine control. So clearly that expectation is not there as a default position. So I think you're right. It's not somewhere you would start if you were making something new. It's primarily a legacy problem.
If it is veterinary vaccine, it's a little bit different than human vaccine because you have the efficacy study that is in target animals. So your in vitro assay would be correlative to the target animal study that you're doing anyway. So if you already had the in vivo assay that's developed for the legacy vaccines, that's a little bit of a different criteria. For products like, like the leptos, if you go back to leptos, in 60s and 70s, when they did the in vivo development, assay development, they had used about, within CVB I'm talking about, they had used about 10,000 plus hamsters to develop that in vivo assay. So they were very well correlated to host animal efficacy. So when we had to go to in vitro, when CVB developed that, we still wanted to go to host animal efficacy to correlate the in vitro. So we didn't make the industries do it, but we did it. That way, each industry didn't have to go to do their host animal efficacy. So we did the porcine and we did the canine. We didn't go to cattle. So the porcine, we kind of, made the leap. If it was protected in porcine and if we could correlate that, we could make the leap. That would be protective to of cattle as well. So we did show the correlation, even though it was legacy in that case. But for the newer products, they can go from efficacy to in vitro, showing the correlation.
Thank you. So I think we can move forward if there is no more question, either here in the room or online.
Yes, Paul Botond, we have a comment from a colleague from Ceva. It's about the preparation of analytical vaccines using different antigen lot quantities, adjuvant concentration, multivalent vaccines to understand better the assays. And in some cases, more than 60 analytical batches are produced. And this says that if the recovery is independent from the investigated factors, they might be satisfied.
Yeah, that's right. And especially in case of multivalent adjuvanted vaccines, the interaction between the adjuvant and the antigens, and also between the antigens, which is called antigen competition, makes the test very difficult. So you can see here that a lot of data should be collected and try to support the introduction of the in vitro test based on available data. So, but I agree with Gita. The pudding's probe is eating. So ultimately, we have to at least at the beginning, we have to make a correlation between the in vitro test and efficacy. But once it has been done, it is not necessary. And if it's successful, then it is not necessary to repeat this in vivo test for each commercial batch. So yes, this is a really difficult, technically difficult, and even if regulators let industry skip the establishing the correlation between the in vivo test and the in vitro test, still we need to make a correlation between the in vitro and the efficacy. So and you can see from Mariana's comment that this may also be difficult. We have to collect a lot of data, and it takes time and takes resources. So resources, already mentioned several times this morning, and I can just repeat that replacing in vivo by in vitro is really a resource demanding task, especially in case of multivalent adjuvanted vaccines. Thank you.
Okay, thanks. Just because it's number 2 on the list, so a lot of people have clearly highlighted reagent availability as a barrier. Does anyone have an experience to share or a specific comment to make? I have some experience from the Vac2Vac project, but I'd like to invite others to talk. Yes,
I
please.
can just make a comment. For some old vaccines, sometimes for some strain, we don't have any monoclonal antibodies, for example. So if we want to replace the method, we have to generate first monoclonal antibodies. Could be tricky.
Yeah, and we experienced this within Vac2Vac. First, we had the challenge to identify the suitable reagents, and that's, I guess it's a little bit more exciting. It's scientific exploration, but what we then had to do, and we only did it really towards the end of the project, was to think about how you make those reagents available, how you guarantee the quality of them and the long-term supply, which is really not straightforward, particularly if we want to arrive at a place where a method can be used by anyone. And so the demand could be quite high. And I think if I was doing anything differently from Vac2Vac again, the main thing would be to think about that element of sustainability of reagents much more thoroughly. I think it is really critical.
Well, for human rabies, we have two monoclonals, different monoclonals, and we set up to have two manufacturers, and there is license between the manufacturers from the owner to the manufacturers. And then we have the sequences also of the monoclonals. So, okay, the tricky point is more the choice of the monoclonals and the making of the monoclonals. For the production, it's a little bit different. I think it's more easier. If there is a manufacturers willing to do that. But we have two for human rabies. So.
Yeah, I think it's relatively easy to solve if if you have your monoclonals identified, you can simply, I think the easiest option is to give the hybridomas to the manufacturers so that everyone can, if the originating party is willing to part with the hybridomas or with maybe agreements, and then every manufacturer can produce their own batches of monoclonals. I think this would be the most pragmatic solution.
Well, it raises the question of the qualification of the batches, but we set up something about that. We have, and we are thinking about the qualification of lots. But normally, if you have discussion between manufacturers and the owners, we should have a licensing process, and normally it works very well. Well, if I remember, for the two monoclonals, it took probably a year or two, but that's normal.
Yeah. I mean, I think your example with the rabies monoclonals is a good example and something that is attainable, but it wasn't easy. It's not easy.
I don't say it's easy.
So it's something that requires
a big hurdle for me.
Yeah, and the other thing I think is important is if you're trying to convince a producer to make these antibodies, you have to sort of assure them some sort of market. You talked about business plans. Well, as a monoclonal antibody producer also has business plans, and if you tell them that will be used by 2 labs, they may need 10 lots a year, and yes, that's all, then they may not be so enthusiastic. So there has to be enough volume behind it or interest behind it that you could convince a producer to do this.
I think monoclonal production is the most convenient for industry if they outsource it to CROs, because there are lots of CROs doing this and professionally doing this, so it's not really necessary for manufacturers to deal with monoclonal antibody production. The qualification is a different question. I think monoclonals should be qualified by the manufacturers using the same test and making a comparative little study to make sure that the replacement batch works as well as the old batch. Yeah. And of course, in case of polyclonal antibodies, it's even more difficult. And the only thing I can mention is that in that case, if you produce a new batch, you have to do a lot of at a high quantity, which would last for many years. So antibodies usually have a good stability if you store them -70, -80, or lyophilized. It longs forever, almost. Okay. Any other comment or question about the reagents? Very similar to the reagents is the references, and it was given almost the same rank. Maybe if some of you have comments on the references, and we can think about international references. There are institutes, there are organizations producing international references. And in that case, I think it is also important to produce a high quantity batch. And for the manufacturers, it is important that they don't use the international reference materials in routine. So in that case, manufacturers should produce their own internal reference materials or control batches and qualify the internal references to the international reference materials. So this is the ideal practice, I guess. So any other comments? Maybe there's a, yeah. Also Mariana from Ceva, functional qualification of monoclonals, also important. The experienced bad consistency, and the manufacturer changed the purification protocol. Yeah, so this is why I think that qualification should be done by the manufacturer, because that's the final probe of okay, and there is one more.
No, I'm not sure about that, because the manufacturer doesn't own the method. So the qualification should be performed by the owner of the method.
But the manufacturers use the method.
No,
so
most of the time you mean the manufacturers of the vet manufacturer, or you mean the manufacturer of the monoclonals?
The manufacturer of vaccine manufacturer. The vaccine manufacturer.
Okay.
Not the antibody producer. Yeah. One more comment from Nora Tereni from Ceva also. So Nora, please. So Nora, turn on your microphone. We cannot hear you. Or maybe you can write it down. I don't know if Zoom doesn't seem to work for the participants, for the online participants, because they just cannot turn on the microphone. Or is there any specific instruction how to?
No, they should be able to.
Can you hear me, Gergely?
Yes.
Yeah. Okay, good. So it works. I can confirm.
Yeah, that's right. Okay. So Nora, please write it down. And I can see no microphone. Okay, so write it down, please.
Botond, if you can hear me, yes or no?
Yes.
I asked the microphone, another microphone, Mariana Jungbaëck from Ceva. The other problem with the reagent availability is that we have to provide them to the authorities, and it is unpredictable. So do you have this type of problem? I think other manufacturers, vaccine manufacturers.
So I missed it. Is it the reagent or the reference material?
Both. So
Both.
the materials required by the test.
Yeah.
Yes.
Okay.
Yeah. It's a problem.
It's a problem, but we can foresee and predict such a request, and usually authorities and governmental labs don't really request high amount of reagents. They just need a quantity which is sufficient for performing the tests they want to perform. They want to control the batches. So yeah, we do that. We provide reagents and reference materials, which sometimes is internal control. But in case of availability of international control, international reference, we can also order for the authorities and send them for the same source as where we order our own reference material. So all in all, yes, we know that we have to get prepared for that, and we have to think ahead and produce or order sufficient amount of reference and reagent materials. As far as I can see, compared to the internal use of reference and reagent materials, the quantity that authorities require is much less. So, and the question from Nora, what if the reference material behaves differently in the given method than the test item?
Yeah, I mean, I think that's a really good point. You know, when international references are established, there is a comprehensive exercise to show that they are fit for purpose, but that can only be done to a certain extent. And it clearly can't incorporate every single product that exists, particularly if we're talking about an exercise that involves animal assays. So this is the reality in some cases, that there is not parallelism between a reference and a product. It's complex. I don't think there's always an easy answer. And I think it's definitely something that we will get into in a lot more detail at the meeting that Joris mentioned earlier, that's going to take place next June, I think, which will really focus on this type of issue and other issues related to vaccine standards. Catherine.
And so it's just some addition to this question. This is a good reason why manufacturers should participate in international reference material production. For example, now a new EDQM BSP project just about to start, the BSP 168, which is about the second replacement batch production of Clostridia, the multi-component rabbit antiserum. So in that case, if you are participating, your product will be included in the qualification and in the preparation of the reference material.
Thanks, Botond. You took some words from my mouth, and I fully agree with you. But I also was going to say, you know, we talked, it came up earlier about what is the value of an international reference standard, and there is this challenge, and sometimes we don't get the parallelism with the test. But we've seen in the past this also, and usually what then happens is there's a in-house reference, a homologous reference that's qualified against the international standard or standard in international units, so that you have a sort of a baseline, an external calibrator. But for regular testing, you would use your in-house where you do get the parallelism and the proper validity in your assays. So there are ways around this, and homologous reference preparations are something that are quite common and definitely necessary in some cases.
Yeah, I think, so Laura has put the link to the standards meeting for next year in the chat, or someone has, thank you. And there's a comment we've received as well from Boehringer relating to the point on comparisons again between in vivo and in vitro, which I think is a really useful comment, so thank you for that as well. Right, I think we're eating into the time for the next session, so we should move on to the next exercise for you all, which is to think about approaches, the best approaches for speeding up development and implementation of methods. And I think we should think about this on a global level, so not just from within one particular laboratory, but what is going to help the most to get methods developed and then widely implemented. So please have a think and rank the options. Thank you. And just a reminder, don't forget to click send at the bottom so that the votes get cast. Thank you, because I just forgot. So we had 39 respondents last time, so we'll just give it a minute to see that number go up a bit higher.
We need to speed up the international collaborative studies then.
Well, so I was just going to make that point that, you know, it's very interesting to see that at the top of the list. I think it's a really effective way of getting people involved in understanding how applicable the method will be to different companies' products. The drawback is that they do take a lot of effort and time, and you know, maybe there's a way to strike the right balance there. I don't know, Catherine, you touched on this earlier. Maybe you want to, again.
Yeah, no, I mean, for me, I think international collaborative studies are very, very important for getting a broad acceptance of the new approaches and to be able to see for the individuals involved in the study how it works with their own products, and they can use the data developed in the study for the validation and regulatory approval of their own products later. And this is actually one of the points in the EMA J3R's paper about how to move from participating in an international collaborative study to regulatory acceptance and how there is an advantage to participating. But the question, I didn't put this as first because the question was speed up. Okay? So hugely valuable, but will it speed up for an individual manufacturer? I think that you could debate. I know colleagues, some of you may know Dean Smith, would argue that the collaborative studies are not the fastest way to get regulatory approval. I think, again, as I said, they're hugely valuable and have a lot of method, but if the question is speed, I might not come down with that as my first.
Yeah, I would like to echo that. I had number 4 as number 1 personally, and why? I mean, I'm you know, I don't debate number 1 and 2 obviously, and even 3. because we can learn from them, and they will foster international sort of recognition and acceptance. But I regard those more than as pilots, really, if you will, hopefully with concrete result. But the portfolio of some manufacturers are so big that you can only draw so much of these studies. So in the end, you have to do it yourself with the products that you haven't tackled via the international studies. So I think, yeah, for me, number 4 is important.
I would argue a little bit, if you allow, okay, because if you do it yourself, that's just the first part of. If you want to help others and speed up the global implementation of the method, then you have to share it, and that's number 3. So if you don't share it, if you keep it confidential, as we heard in some of the previews, probably by Gita, if individual companies initiate a new method and keep the information confidential, then it doesn't help in the global implementation of the new method. And so the individual company may even protect it by a patent. This is a real life example. I know at least one such an example. So when we try to implement the same method, we are blocked by the patent. So this is very, very, very important and very serious roadblock if a method or just part of the method is patented. So I think we should change our mindset, manufacturers. And of course, it is understandable because a company is a profit-oriented company and wants to have a competitive advantage. So this is why always companies try to protect their intellectual property information. But in this particular case, when we have common interest, common goal, I think we should make exemptions. And in that case, companies shouldn't protect and shouldn't keep this information by themselves.
Maybe you need to, I think all of these ideas are good, and we're getting to the point where you put these all together, where you need private company investment in multi-stakeholder projects so that you can share case studies and then do collaborative studies. And with all of those things in place in the right order, it will be fast. Yeah.
That's a very nice way to summarize it, Catherine, thank you. And there's a comment from Dr. Tiber online as well, referring to the fact that if we can identify the specific area of collaboration and then agree in advance that sharing will be part of that collaboration, then that can be a very effective path forward. And that's essentially what happened with Vac2Vac. It was a very effective means for sharing not only materials, but information. So, thank you. Okay, I think we will move on to the final question, where Laura would like everyone to identify which current method that uses animals should be prioritized for development and implementation of an alternative. And I believe that the responses will generate a word cloud, so that the things that get mentioned the most frequently will appear larger in the cloud, I believe. So have a think and describe or state the current animal method that in your view is the priority. Thank you. Okay, thanks. I think probably some responses are still coming in, but there's a clear theme developing: rabies and Clostridia. I think probably the two commonly expressed priorities there. Multivalent products, because you can target one antigen and it has no impact if you're using animals to assess the other antigens. I think that was touched on earlier as well. There is no cloud, Laura. No, just an old-fashioned list. Okay, lists
But we can see.
It can still be useful. Yeah. Okay. Does anyone want to comment specifically? Share any experience specifically? Kat.
Maybe just no surprise that the rabies one comes up there. I think, you know, the good news with rabies is in Europe that 3 of the manufacturers I know of all have registered glycoprotein tests and stopped doing the challenge tests. We have bigger challenges internationally, and I think maybe this gives us some more ongoing discussions with the US. I know things have stalled a bit. We've had conversations with the USDA, and I think that's the next big target. And I know that the human BSP rabies project that came out of EPAA is moving forward well, so that we maybe have some further opportunities to discuss with Jean-Michel whether we look again at that for the veterinary vaccines. Obviously, the antibodies there are well characterized. They're being moved forward for human rabies vaccines. So I think it does present some opportunities there to look for some further collaborative work on the front of the veterinary rabies vaccines outside of Europe. At least, you know, we have companies who move things forward in Europe. Whether we relook at Europe is another story, but I think it does present an opportunity for potentially some collaboration with the US and working with the USDA and seeing where companies think this maybe could go, if nothing else, just to start by testing the antibodies to see whether they will work for the veterinary products that are on the market in the US. If that then at least gives us a starting point, it's certainly something. And the Clostridia makes me think that maybe that's a nice targeted vac-to-vac follow-up project, Imke, on just Clostridia. I know there's work going on in different, you know, people are doing work on Chovii, some work on Septicum and Perfringens, but there's still a lot of Clostridia, tetanus obviously, but there's still other Clostridia to hit and still some progress to be made there. They come up, I think, in every survey every company does internally. They are top of the list of things that people are concerned about us moving forward.
Okay, thanks very much. Thanks everyone online for expressing your preferences and for the comments that came through as well. Thanks everyone in the room for engaging, and we'll move on to the next session, I think. Thank you.
Thank you.
Thank you very much, Paul, Botond, and all of you. We will make sure to capture all these comments because it might be useful for further discussions and potentially understand if there is the possibility to work more together as we have been doing today in setting up something concrete or in parallel approaches, because as we have been seeing, there is just probably not one approach that is going to allow us to solve all those issues, as Catherine said. So we probably need to make sure that all the available opportunities are going to be taken with the right stakeholders with the right timelines. So thank you again. We are going to use the same concept, but moving more in the direction of a more focused discussion on the regulatory opportunities. So our moderators for today are going to be Milne from EDQM and Catrina Stirling from Zoetis and HealthforAnimals. So same concept. So if you want, we have a first question that can be used to kickstart the conversation.
I think that makes sense, Laura. Let's kickstart the conversation with the first question. So we're looking more here at the regulatory side of things and how we accelerate regulatory acceptance. We've touched on a lot of this morning, I think, with sort of various different aspects of it, and it came up actually yesterday as a key theme when we were talking broader than just vaccines at the EPAA annual conference around the general how do we speed up? And the questions from the politicians are, how do we do it faster? What do we need? Do we need investment? Do we need, like we say, collaboration, engagement? And I think the message there again is all of those things, but definitely to look for opportunities to be able to do it. So let's see what everybody thinks from the first Slido question. It's interesting watching this real time and how it changes. Can you tell how many people have responded, Laura?
Yes, just look at the top right.
Is that top total? Okay.
Yes. So 9, 10.
Okay, we'll give everybody a little longer because I know there's a lot more than 10 online. I would say to everybody online, just go for your instant first reaction. Don't spend too much time thinking about it.
That's what you mean by regulatory resilience.
That wasn't my terminology. Should we see if we can get to past 30 people responding, Laura? Is that that's probably a good sample. We give them a couple more minutes. I was going to say, is everybody on the room connected? Everybody in the room connected? Have you scored? One more. If we get to hit 30, let's see if we can hit 30 and then we'll, there we go. Okay. Catherine, maybe a good time to start having a conversation now. Yeah, so it's getting close at the top. Thanks, Laura. So it looks like the top and probably exactly the sort of things we might have expected. Is that mutual acceptance, reliance on data between authorities or similar approaches, and early engagement and dialogue between industries and the regulatory authorities. So I don't think any big surprises there. Precise guidelines is an interesting one. There are times when, certainly from an industry point of view, I see this personally, where precise guidelines are not always something we want because we want the flexibility, but at the same time, I think when we think about global international acceptance, precise guidelines are of value in certain contexts. I think that the challenge is always that to have them there to help facilitate acceptance, things like monographs with fixed methods in them, etc., that define things very clearly, versus the fact that you don't necessarily have to. I think we still need to maintain that as an option, that you can follow that precise guideline, or you have the option to do something different if it works for you individually as a company with the right regulatory acceptance. So that's data sharing is an interesting one. And we've talked a bit about it, and it's data sharing. What does that mean to people, and what's the expectation on data sharing? It's one thing to do it within the context of a collaborative project, but there's always this level of concern from an industry point of view of sharing specific data on your product and the specifications for your product in an open way with other companies. It's one thing sharing it with a regulator or sharing it in a confidential way with a third party. And that's something actually that happens quite a lot through some of the EPA projects, where And NC3Rs, I know they do the same thing. They act as a sort of data broker so that companies will share information and then it becomes anonymized. So you don't necessarily have then that direct relationship to an individual product, but it allows the regulators to see how things look across a range of products without the industry concern of you can identify whose product's whose. So data sharing is an interesting one. I guess my question on data sharing is, what would we be hoping to achieve with it? What's the aim and what are you trying to do? Speed of regulation review process, that's very much with the, and regulatory resilience. Laura, what did we kind of mean by regulatory resilience?
There is this new concept that is coming up also within the more stringent regulatory authorities, where the concept of resilience means that the different regulatory authorities are relying on all the mechanism of those more stringent regulatory authorities. So they just don't do the overall the batch release testing, but basically they are fully there is the reliance and there is not only the reliance concept, but also there is the resilience in the fact that they are going to be flexible in the way they are going to accept or they are going to modify their pharmacopoeia's requirements. So they are not going to write the specific settings, but they are allowing the possibility to change, and they just provide guidelines on the way they are expecting a new methodology, new testing, or new products to be produced and tested.
So I don't know if everybody heard that, but Geetha was just saying that we're more kind of almost talking about regulatory flexibility. So that maybe goes a little bit to what I was saying about precise guidelines versus, the concept in the pharmacopoeias of you can use any suitably validated alternate test accepted by the regulator. You don't necessarily need to follow the exact method that may be precise in a monograph, for example.
So they're kind of contradictory, isn't it? Then when it comes to precise guidelines. I mean, one of the reasons US stopped putting out this standard requirements was because it's so restrictive, but it at least provides direct pathway. But to give the flexibility, we stopped that. So it's kind of,
It is, and it's this double-edged sword is that there are certain situations, and it's probably more with this difference between the global innovator companies and some of these smaller companies in individual countries, where for them precise guidelines help, because it's okay, that's accepted, that's what we do, that's what we follow, don't need to think about it, we know if we do that, that will be accepted. At the same time, some of the innovator companies, for different reasons, we may want to have the flexibility to do something different, providing that we follow all the same registration criteria. And I don't think they necessarily need to conflict. I think that both options are valuable and they need to be there. They provide a different route for different stakeholders. But the precise guidelines are something, particularly when things go into a pharmacopoeia monograph, etc., or something's published, or you know that that's accepted, whether it's in a memorandum or something else, it does help facilitate that international regulatory acceptance, particularly if there's less experience or less confidence with a certain regulator in terms of that approach. If they know that, okay, that's accepted, if something goes into a WOHA guidance or terrestrial manual or pharmacopoeia, etc., then it's easier to follow. But if you're dealing with something where you want that flexibility, and I know you probably get conflicting messages from industry. It's like on the one hand, people say, well, we want precise guidance, and then the other hand, people say, well, we want to maintain the flexibility. So you're probably hearing kind of mixed messages. But I know, Catherine, if you want to.
I was going to say, I think that you could say that exists to some degree already in the pharmacopoeia, and from what I understood from your presentation also in your guidelines, you have the monographs, and in the pharmacopoeia there's the general notice that says if you have another test and you can show that it's a valid test, and you can do that. So that in some ways exists already. But that's why I'm not entirely sure what the precise guidelines means you want a step-by-step, this is exactly how you should do the test and this is all we accept, then I have I'm reasonably certain that there will be a lot of objection to that for the very reason that you say that you need, in some cases, to have the flexibility.
Well, when we do have the standard requirements, codified requirements, then the flexibility is limited. I mean, some tests are different, but if it is for efficacy studies, for example, if there is a standard requirement, giving exceptions to that is harder. Because by law, you gotta follow the requirement there.
So that's maybe a difference in how things operate in Europe versus the US. And actually, I've seen that conversation talking about developing VICH guidelines with the way in which the regulators in Europe would look at a guideline versus the US regulators. And I've seen that heard them say to me, well, once we've put it into a memorandum or something, or the VICH guideline, then that's what we'll follow. Whereas the European regulators often take a bit more of it is a guide, it's a guidance, it's something that can be adapted on a case-by-case basis. It provides a guide, and if you follow it, that's great, but deviating from it isn't necessarily so much of a problem. And I think that again goes to that sometimes that fundamental difference in approach, which has stood out to me something when we've been talking about VICH guideline development. You see that in the conversations.
Yeah, I mean, when it comes to that, changing the standard requirement or CFR codified requirement with VICH guideline development, it was possible because it was the harmonization criteria that we bought into. So that's why, because of the international collaboration, that was possible. Whereas if a company comes, your company came on its own and asked for, say, target animal batch safety exemption, if we weren't in the harmonization, arena, we couldn't have made an exception to one company. So that's what I mean, having the international collaboration helps that pathway.
Yeah, I'm just going to make a comment in terms when I look at regulatory resilience, having worked in regulatory agencies, I read that as a regulatory resilience in terms of over the years, the layers of confidence the regulator has in the quality of the product enables them to make a risk-based approach to say, okay, we may not have all the data here for entirely to be confident of a completely validated test, but we have other issues. We have we have the pharmacovigilance, we have our GMP inspections, we have our, other systems of now that with big data and the way of actually getting information back to the regulator is speeded up with all the IT systems we have in place, we can make those decisions. We're a resilient organisation. So I sort of separated regulatory resilience from also regulatory flexibility. And when I see the time-precise guidelines, again, having helped develop some of the guidelines at the European and national level, I think the industry don't see guidelines as precise and sort of almost mandatory anyway, from my experience. They don't really offer the flexibility. And we've recently involved in some of the VICH discussions involved in the Royal Terrestrial Manual on the project we're being involved with, and it states that for the preclinical test you have to use 8 animals for every target species in each of the categories, and then people assume that's written in stone, but if you read it, it does offer the option to do other numbers of animals. But what I see all the time is the industry will just do the standard VICH test. So I think precise guidelines, I think we do need flexible guidelines. I think, definitely need flexible guidelines. And I think the resilience there, I'd say regulators do need to work together in sort of sharing their understanding of the risk they're willing to take in authorising those tests. Thank you.
Maybe guideline is really misleading here. Maybe it would be better to have a kind of checklist what should be provided, or a procedure to follow to gather all the things together you need for a certain authority. I have no clue, but when I see the discussion around guideline, the wording of guideline, I think this is misinterpretation here.
Catherine? No, let's, if you know, do you want to jump in on something else? Go for it.
Turn my mic on. I was interested to see the number one was the reliance and mutual acceptance of between regulatory authorities, and I was curious as to what the responders understood by that, because I mean, you know, in the EU, we have, as Kat mentioned earlier, there's the official control authority batch release for a restricted list of products where testing is done by one national control laboratory, an OMCL in the EU. A certificate is given, and all of the other countries who require OCABR and who use the product in the EU will request that, will receive that certificate, and it's accepted. This is based on a legal background within the now regulation for veterinary medicines. It's possible for other regions, countries, to also recognize these certificates if they choose to. That wouldn't be mutual acceptance, it would be reliance. And we try in the EU system to be as transparent as possible, because that's important for reliance to understand what you're basing your decision on. So there are guidelines that are published that tell you what's being tested and protocols, etc. But I was curious as to how many other countries require this batch-to-batch testing for batches and would potentially accept certificates, and if there was something else behind this response, because you could think wider in terms of regulatory decisions and reliance on regulatory decisions. So maybe if those who responded could tease out a little bit what they were thinking in responding to this question, that would be helpful.
Yeah, I mean, I read this more looking at the acceptance of the method from a licensing decision or a regulatory decision point of view, as opposed to the testing of individual batches. There are networks of regulatory authorities that are operating on this basis. I mean, obviously the European system is a good example, but there are others, and it's a potential for a quick win, a rapid acceptance and dissemination. So that's how I was viewing it. It was about the data and the acceptance of the regulatory decision.
Does anybody else want to comment on that, or anybody online? If you don't want to speak up, feel free to type a response to that question in the chat box. David.
Yeah, I think I was answered that one from a very similar point of view of Botond. I think I may be, as industry, we're mistaken on this, but our feeling is if we go as industry to a regulator and say, we've got this great new alternative method, yeah, okay, we'll look at it, things. If another regulator comes to you and says, we've got this new, non-animal method, it's got a sort of more credibility. You take more notice. I don't know if that's true or not, but that's certainly our feeling.
So I mean, for those people around the room, so I think this is would indeed help to speed things up. Are there mechanisms that exist already between the authorities? I'm not a regulator. As Paul pointed out, in the EU, if you go through the central authorization procedure or the MRP procedure, the DCP procedure, this is more or less what happens. It's a combined evaluation and decision. But are there mechanisms outside the EU or between the EU and others that this would be possible?
The only example that I would have is we do accept some of the data, not as complete data for licensure, but say, for example, if you're doing your pre-license safety study, which is a field trial, as a requirement with the USDA CFR requirement is three pre-license studies in 3 different geographical locations within the US, but we exempt one of them if that has already been done, say in Europe, and we can use that as one of the 3. So that's an example I can give. So for licensure itself, we don't take the whole package, but as supportive data, we can.
For the mutual acceptance as data, there's obviously the mutual recognition in there in the European from an Australian experience. I think they have a really good example because they don't have really the technical requirements in legislation. So they have a sort of quite a complex legal framework, but they don't have a technical guidance. It's based on guidelines. But the approach they've taken is that they will recognise both European and US requirements for registering vaccines. So for me, that's a good example of a mutual recognition of data. It's a pragmatic decision saying, okay, there might be different standards between the regions. We will accept products without with the sort of fast-track procedures to make sure there's no critical issues for the Australian market. But it is a really good approach, I think, in saying, we don't really see a safety or sort of efficacy issues with either region, so we're quite happy to take both, and I think that sort of facilitates the sort of the availability of products on the market as well. Thank you.
And I guess the other place that mutual recognition exists outside of the registration concept is with GMP. So there are now mutual recognition agreements in place between the US and Europe on the human side. I know they're still working through the GMP MRAs for veterinary products with FDA, at least as a starting point. And those exist with other countries, under the context of the PIC-S schemes and when everybody's working to that similar standard. So, that principle is already happening in other types of areas of medicines regulation. So maybe it is an area for further discussion in terms of broader, as you say, acceptance, but then maybe then goes to sort of using, so for example, if you think the OCUP testing, or is everybody using the same sort of test? So if something's been done in Europe, is that certificate suitable for multiple countries around the world because that's the same product that's been tested to a same level that's recognized internationally as a suitable test to confirm that it's acceptable?
And are the specifications the same? Because globally you have different specifications. So even within Europe with older products, we have this problem because the specifications aren't the same. But I think if there were my experience is to get mutual acceptance, because in the OCABR network, and it's not the regulatory, but what you need is you have to start with mutual confidence. I think the Australian approach is very good, but perhaps I don't know if it's unique, but unusual in that do they have interactions with the EU authorities and the US authorities to understand how the decisions were made, or is it just an acceptance? Because my experience has been that to be able to trust someone else, you have to know what they're doing and how they do it, so that you can say, well, I would have made the same decision.
No, certainly we've received the dossiers, and then again, it's the critical to make, again, it's similar to make sure that the product is not going to cause any harm to the animals, the environment, or to the public. And I think those sort of three criteria are sort of protected, and also a risk to trade, because Australia is a big trading nation. So once those four criteria are met, then that product would receive a registration in Australia, either whether it comes from 9 CFR standards or European Farm to Feed standards.
Maybe just to add to that, and I can't recall exactly if it's happened with Australia, but there have been scenarios where we have actively shared European assessment reports with agreement from the agency with other. I know we have examples where we've done it with Canada, which has facilitated faster approvals. And that, to your point, Catherine, and in those scenarios, I think they did actually have some conversations. We weren't part of it, but we just made the introductions, we shared reports, and we said, yes, you can talk about this product between the two agencies. And that did facilitate that understanding, and I think it probably does help the regulators in whichever country it is and understanding how that decision was made in the other region. And that's doable because we have these formal assessment reports we get in Europe, that can facilitate sharing and then further discussion, but it also, which I don't think you really have in the US in the same way, because the European assessment reports is sort of everything together, and you can see where the regulators' thoughts have been. This is what the data are, and here's our opinion of it. So it's And you see a lot of that even in the public assessment reports once they're published. So there's quite a high level of transparency in Europe around how the decisions have been made. I think necessitated by that public request for transparency. How did they make the decision that that product was safe? But it does mean it gives us the opportunity to share a bit more of that regulatory thought from the regulator side with other international regulators. And you probably don't have it quite the same, but you know, it's doable in the context of acceptance of a batch potency test because we have an assessment report. I don't think we've ever done it, shared that internationally. We've only ever done it with new products, but it's an interesting thought if that might help facilitate assessment, not just providing the same dossier, but also consideration of providing within regulatory authorities that assessment of how the European thinking has been, because we've certainly seen it help with new products.
We've had sort of a Euro-US view of things. Are there any people online or elsewhere in the room that would like to comment on the possibilities for this?
Everybody online is very shy. You can type them in the chat or comments in the chat if you support what we're saying, if you think it's a good idea. Carmen.
Yes, I'm a dinosaur. I learned it again. We had this awful review of existing products in the late 90s of the last century. And all authorities were really under an avalanche of applications. And at that time, with the allowance of the manufacturers, we spread our assessment reports to the authorities who asked us to do so. And there was agreement on all sides, and it was so simple. The vaccines were already licensed everywhere, and nobody had the time to make all the reassessments. And so we helped each other informally, and it worked.
I was just going to ask a question, you know, when you called on Asian countries. From my understanding, majority of the poultry products go to Asian countries. So when you register the product, do you develop more data, or do you get more data from those countries, or do you use the data that you obtain from either EU or US? I'm just curious. This is a question for industries like you.
I'm going to ask Marianne in a minute, but I don't get involved a great deal in those kind of geographical expansions, so I'm only going to speak from a general point of view. I think generally speaking, we would use the US or EU data sets for international markets. We may, if we know there are specific additional requirements in some countries, I know there are some for China, but for the majority of markets, and there's some specificities with Brazil for stability requirements. But generally speaking, we would use either the US or the EU data package, depending what we were doing for most of the international markets, with a few exceptions. I don't know, Marianne, if that would be the same, or Corinne.
Yeah, that's the same.
Yeah,
we submit the same package globally with some additions for specific requirements. And it helps to be able to share the assessment reports from EU authorities with non-EU authorities, and not particularly Australia, but even the smaller countries. They ask for the EU approval, and that facilitates the acceptance of the variations. But that's not only for going to in vitro methods, that helps for a lot of variations.
I agree, it's exactly the same. So.
Additionally, you know, with regard to the data package, for EU and all, you submit the whole package. Whereas with US, it's piecemeal. Does that cause hindrance when you're collaborating with other agencies?
So what we would normally do if it was a US-based product, where often we don't have most of our products are either US or Europe, and then they go to the rest of the world from one or other region. The European dossiers are certainly easier for everybody, because you say they're a whole package. The US ones, we would normally license in the US first, and then we would put all the pieces together for the international dossier. But I think, I don't know, it would be interesting to hear from like our colleagues from Indonesia, is if you've seen a US style dossier from a USDA product or a European one, how you find them, because I certainly from experience, I would say that if it's a USDA product, there's not the same tying everything together, because like you say, you assess it piece by piece and FDA do the same. So it doesn't kind of bring everything together in the same way, whereas Europe, we have everything together and then you have an assessment report that covers everything. So I would speculate that international authorities find the European dossiers easier to digest because they tell more of a story. But the reality is that the majority of the products that we have internationally are probably more coming from a US-based approach. So I think a lot of the international authorities are quite used to that format of how, just having bits of the US put together into one. But I'd be interested to hear from our colleagues from Indonesia.
Okay, regarding your question about the, maybe you can repeat your question. Yeah, okay.
So when it comes
data.
Sorry?
Yeah, maybe you can.
When it comes to data package.
Okay, data package.
When the
yeah.
industries are submitting.
Okay.
The package, ours with the US, it's kind of a piecemeal.
Okay.
So we review each study on its own independently, and the industry actually has a tough time putting it all together.
Yeah.
Unlike the EU where they review the product, it's a whole package they review. So which one is easier for both for the industry and the regulators from other countries? Which is preferable? That's the question.
Yes. From our side in Indonesia, when the one products come to Indonesia, they have a whole package of data and information as our requirement with the regulation refer to the administration and registration. They have a complete of the data package, including the from the composition until how they implemented the GMP, and then in the one other side, they must be explained about how they testing the finished products, how they identify raw materials they use in the production, and then how they can determine of the like toxicity, potency, safety, and all of the data must be submitted to our government. And then we have a commission for the review all of the products come to Indonesia, and then regularly the products must be reviewed and presenting in the front of the commission, something like that. Yeah.
I think, Geetha, sometimes what also happens is that our colleagues locally will take the pieces of the US information and put that into a format that's required locally, sometimes translated into their local language. So they do sometimes put it into a different context. Any other thoughts, comments?
Yeah, from Ceva, I can also confirm that we either use US or Europe data. However, we face more often the differences in interpretation. It's not specifically potency, but like safety or efficacy experiments where as part of the Europe assessment, we conclude we we may not need to perform all experiments because we have the scientific benefit risk-based justification. However, we have examples it was not accepted with a specific country, and we had to perform that experience, whereas it was because of a different interpretation. And the other is additional experiments or local testing to run repeat release testing in countries. So these are mostly the experiences on that.
Thank you, Gerli. Catherine.
Thinking about something that Sonia said earlier, and I don't know if she's still online. I think it's still over, but she talked about the EMA group starting up training for EU assessors, and I wonder if that would be open more widely, because I think training, you learn, but you also exchange, and it's a way to really see how others think and maybe get on the same page with respect to what's needed, what's not needed, what's essential, what could be flexible to them in the case-by-case aspect. So, as I said, I don't know if she's still online, but maybe if they get this program off the ground, if it could be spread to a wider geographical area, then that could be interesting.
It's interesting, Laura and I were talking about just that thing earlier, and the concept of training, and the need for, meetings like this are useful, but actually some of that training, more detailed thinking about the regulatory approach to assessing that data, the expectations outside of Europe, with some other international regulatory agencies may be very valuable. And we were just trying to think through how you facilitate that, because one of the challenges with that is often internationally is just getting the coordination and the resource to coordinate and to get people there. Because I know even just setting up this meeting, it's difficult for regulatory authorities to get the funding to travel to something, but I think we've got many more opportunities now with everybody's, maybe it's one of the upsides of the pandemic, everybody's level of comfort with Zoom meetings. You have a much better chance of being able to run something, and people are more comfortable with running these kind of training sessions, although it doesn't always substitute for that face-to-face interaction, but at least you've got an opportunity to use those kind of technologies now. Laura, do we have any more Slido questions? Let's go for the next one then. Yeah, so this one is, what is still missing at country, regional, global level to make new methods acceptance a concrete reality? Is this a free text one? Okay, so put in make it a 1 word, 2 or 3 words, whatever you feel like. What do you think is missing particularly? And think about your country locally, your region, or what you think might help on a global level. Can we do a word cloud from these, Laura? It might be interesting to, it's doing it this time. Now it should work, okay. I can encourage everybody in the room to do this too, if you're not already connected to Slido.
Do we leave this open?
I was going to say, should we think about starting to, maybe let's leave it open for a bit longer, Laura, but maybe let's start talking through some of them. And the first one that's right now is the biggest, is what we were just talking about earlier, was training. And I'm guessing for people, that's very much training for assessors, training for agencies and helping them understand new methods, how people are validating them, how to go about assessing them. Dialogue and trust, we've talked about. Method harmonization. It's an interesting one, method harmonization, because I think we've talked about this earlier of industry wanting a level of flexibility, but at the same time, I know that for the regulators, method harmonization is easier. And for me, that's one of those areas where if we want method harmonization, the easiest way to do it is through collaborative studies, because otherwise the industry won't work together on those kind of things, because we have our own individual projects, and many of the products, there's so much variability between products that method harmonization isn't always a viable option. But there are certainly how shall I say, there are examples out there where harmonized methods work. But I think the other challenge you have is in many cases that the variety of strains, even within one type of product, there's so much variability that it's difficult to have what I would call a truly harmonized method where everybody's using the same method with the same antibodies. I mean, what they're doing on the human rabies is a nice example of where it should work. But if we think about then other things where people maybe have a lot of variability or different strains that certain reagents may not work, getting reagents that work across everybody's strains, everybody's isolates, it's not always an easy thing to do. I guess it's, we can have harmonization on the principle of the method, of the approach that you can use, say, like for Leptospira, ELISAs against the LPS antigen, that is a suitable, but you then need to develop individually. If it also helps if the reagents are commercially available, they're not proprietary, because then other companies can, if you know that those two antibodies have been used for that, then you can test them, try them. But method harmonization is an interesting one. But I'm trying to look at these and thinking in terms of concrete actions from this meeting, Laura, that stand out. And I think method harmonization I'm going to stay away from, in the sense that we've talked about projects. There's projects that we have running, the BSP studies, etc. They're good examples, Vac2Vac, where we're trying to move those forward, and there will be other opportunities in the past. But dialogue and trust and training stand out to me as ones where maybe it is possible to think about some concrete activities. And I think they certainly look fairly consistent, and I don't think ISA and Sonia are still online, but they're certainly consistent with the EMA 3 Hours Working Party, some of their aims and agendas around that, training being a big one. So I think there's certainly room for some further activity and discussion on that front. And through things like training and meetings like this, you build trust. So I think they probably go hand in hand. There's a lot of other interesting things, but training and trust builds knowledge also. And I guess that training also can help build pragmatism and mutual recognition. So I think that does stand out as one of the, and regulator exchange, it stands out as one of the main things that might help facilitate all of those things. So it's maybe a pretty good reflection of where we're at, but that's my opinion. I'm going to see what everybody else maybe go to my co-chair first. And Catherine, thoughts?
No, I mean, I think as far as what we can get out of this meeting, the idea that maybe finding common basis and training and what to look at when you're assessing a method, because as we've said, method harmonization can come through a collaborative study, but you could also have methods arriving independently, and if you don't have a lot of experience with those methods yourself, to assess it would be, I imagine, quite a complex task. So having networking resources amongst the authorities in particular, but also, I mean, the manufacturers have associations, you can network and try and find ways to work together as far as possible within your own context. But yeah, I think that could be something to take forward from this group. And I'm definitely for method harmonization, or at least the one collaborative studies where where we have a good way forward. But I think this is an important exercise, and we've seen what's still missing, but it says, what can we do to make method acceptance a concrete reality? And I think that we already have it as a concrete reality to some degree, and there are good examples of it. So maybe it's more a question, how can we propagate this rather than say that it's not there yet? Because I think we do have some good examples, and we can build on those examples.
I agree with all the comments. I think I've been in a few regulatory agencies, even the big ones, they often lack, even with the experienced assessors, the experience in sort of dealing with some very complex variations to change from an in vivo to in vitro test. And it was always easier if there was a sort of in vivo test you had to correlate. I know the problems associated with that, but for an assessor who's actually then trying to make that link, it's easier if you take that the requirement out, which is great, but it does make life even more complicated. What we've done in the past, we outsourced to some experienced statisticians as well in the area, because we didn't have that resource in the agency to do it. So I think the training initiative, bringing in, because it's not just one assessor here, you need a team actually to do that sort of work, and often the regulatory agencies don't have that resource or experience, and that's why I put prioritization, because often 3Rs takes us a step back. And it was interesting to look at the EMEA last publication, the biennial report with 2016 to 2017, because other priorities took over. So I think training is fundamental. I think bringing in the right expertise to those agencies to support them, and a training, broad training, it would be a great initiative, I think, to get a better understanding of what the companies are doing, because the regulators are often a little bit behind to get that experience. Thank you.
Thoughts? Anybody on the phone or online want to add anything to that? Does that make sense for your region, David?
Yeah, I was going to sort of postulate, I'm certainly working in the association. It's been more than 20 years since I was in the lab. And I know things have moved on a long way in terms of lab techniques since then. And I sometimes find myself in a meeting thinking, what are they talking about? And I'm wondering if that's also the case for some regulators, where they're just not familiar. They've been in the office, they're not familiar, if they're not working in the lab still, with the latest sort of scientific tools and techniques, and maybe some basic almost training in this is what this technique is, this is what it can do, this is the sort of problems you can get with it. And that would sort of help build some basic level confidence in some of these new techniques we're talking about.
And David, to that makes me think that actually it's also some basic level understanding of how industry, for example, is going about using the consistency approach to set specifications. What is the process, the thought process, the technical process we go through in deciding how we put together all the information. And I know we maybe don't always do the best job in our dossiers of explaining what we've gone through in terms of that process. And there maybe is also in, not just with the methods, but actually the way industry's going through the process of defining those specifications, setting them, how we've used we do use statistical analysis, trend analysis, standard deviations, to say what's the variability level, what's things like using failure analysis and that sort of thing. We use that all the time to try and assess what's going to be viable for us to set specifications on, so that we're not going to have batch failures because we've got a more sensitive method. But there's a lot of different techniques that we use as part of that development process, and maybe that is another area for training that would help. We've certainly found in other things, it's helped to be able to convey to the regulators that we haven't just proposed something by picking it out of the sky and gone, okay, well, that'll do for a specification. We've actually gone through a very structured thought process of making that assessment that includes risk assessment and a structured way of doing it, that it's not always easy to convey in the context of a regulatory submission or just providing a report that you don't fully understand. And that's maybe another area for training that would help assessors understand how we go about doing that. Isa, I've just seen you come online. Did you want to add something?
Yes, that's exactly 2 points I was also thinking about, to have, on the one hand, a collection of cases which might be problematic, to discuss them, and on the other hand, to have a collection of examples how specifications were set. Here I would suggest that we make use of the existing VAC-to-VAC network we have. And maybe we could base this collection of cases on the experience from the former VAC-to-VAC members, and of course also additional members who would be willing to contribute. And if we have this collection of cases, our discussions could be much more concrete, and then certainly would help to have an exchange of experience and also a building of knowledge.
Yeah, it's a good point in terms of case study examples. I think it's certainly easier for people to see with an illustrated example, and I guess VAC-to-VAC is certainly a good place to start, and I guess that maybe is a challenge back to us at industry. If there are any particular case studies examples, we would be prepared to share. I mean, I think if it was simply one company was having that conversation with the different regulators in a confidential setting, it wouldn't be too difficult to do. It would be more of a challenge with an example if there were more than one company engaged with that, but we could do it on a case-by-case basis. But there probably are also, in the context of this, maybe some situations where we might be prepared to share, including with other companies, how we've gone about it, because the other thing you do get from sharing experience and how you've gone about doing it is you may learn other ways of doing it. You may think, okay, that worked, or we did it differently. And yeah, maybe that's one for us to think about within the association. If companies do have something they'd be prepared to share in the context of case study examples of how we've approached specification setting with something that we wouldn't have any confidentiality concerns about sharing. I'm looking at Imke, but we're thinking about that one, Isa. And Paul.
Couldn't that be blinded in a way, so that it's only their freedom to share it?
Sorry, Isa, we didn't hear you very well for that one.
Wouldn't that be blinded in a way, so that it's not a problem to share it?
We could think about something like that, maybe with the support of the industry association, of giving some examples to David that he could put into a format that didn't necessarily identify the product or even necessarily the antigen, but it gave you an example. Maybe we could think about it, Imke.
Yeah, if necessary, I would think, because you also want the dialogue, right? Yeah. With the asset developers and his reasoning why he went for this or that specification or approach, so that if we do it, I think that would have added value.
Yeah, and I think there probably are some examples with existing products, particularly where it might be possible to think about ISA some of those examples. So point for further discussion, but I think Paul wanted to jump in.
Well, just to extend the discussion on that point, I think what I would have found very useful in terms of developing a method is to get a really good idea of how far do I need to go to convince someone that this new method has an appropriate level of discriminative power. Because you can do all sorts of things to the vaccine, but you might not need to do everything that you can conceive. It might be nice to do in the lab, but do you really need to do it? So some kind of guidance, some examples on not just the bare minimum, but what is it really that is going to convince an authority that the method you have is fit for purpose, you know, above the normal validation criteria that you would do for any method.
And Isa, that's maybe one back to you a bit from a 3 Rs working group, because that's true also from an industry side, is how far do we have to go? What is the sort of minimum things we need to be able to show, and what type of data would be sufficient for us to do it? And hopefully the regulators now have seen more examples from industry of how we're doing it and what we're doing that maybe could lend itself to some sort of guidance. I know we were talking about guidelines earlier, but maybe that is something that's worth discussing further, I say, with a 3 hours working group.
Yeah, spontaneously, I would say that this is to be decided on a case-by-case basis, and that was also on the slides. I think beyond validation, it's very difficult to give a concrete guidance there.
And I guess that's one of those areas where we as industry, the more we do, the more we learn. You know the questions you get, that you know that you're always going to have to. Forced degradation studies is one thing that comes to mind, being able to show that your method will always pick up something when it's no longer functional. But yeah, maybe it's something to think about, how we could do something a bit better there. Are we getting short on time, Laura, I think? Do we have one last question? That was the last one. Okay.
Maybe just one want to add. We had lots of discussions in VAC to VAC about altered samples, and I think we found quite good concept, and that would also be worth, I think, to share.
Yeah, I think anything like that is going to be helpful generally, and if something's coming out being published from Europe, then it's useful to then be sharing internationally. I know that's something we're trying to do more and more, is share with our international regulatory colleagues within the company reports from meetings like this and that sort of thing, that just helps dissemination of information. But since we need to wrap this up, I would say the big thing that's come out right now from this discussion is the training. Training to build trust and confidence. If I wrap it up in just a few lines. Who, Laura, is wrapping up? Am I handing back to you?
Yes, thank you, me, Joris, and then a few final remarks from the three organizations that have been set up these events. We have seen today that we are on an important moment because of the major closure of VAC to VAC, with a lot of lessons learned and probably many additional results that will coming through with new publications. We have seen that there was a very important jump in terms of the regulatory authorities in having a better understanding about the opportunities that the implementation of the 3Rs might bring in, starting from all the work that Europe has done, following by the work that is also ongoing within the US, with the hope that there is going to be even further collaboration between those two, and the fact that we have seen that other countries and regions are also moving in that direction. So everything that came out also through the discussion this afternoon, it will be important to find a strategy that is going to put together all the current possibilities that we have in terms of facilitate the method development and validation through the different tools that we have, starting from the international collaborative study, big programs like the BSPs or the VAC to VAC example can be definitely further considered. At the same time, it's also important to find out some practical solution about the reagents and how to better and the standards and how to better handle this part. And last but not least, also leveraging a little bit better all the regulatory opportunities and mechanisms that we have been seeing that do exist. And as you were saying, Kat, to try to find a way to have to bring together all the knowledge that is available within industry and the regulators that have more experience in this field, and making sure that this knowledge can really be further disseminated through maybe something that is going to be structured and led by many organizations. We see how important it is collaboration. So it was our first keyword this morning, and we come back to that. So the collaboration between the regional, local, international authorities and other organizations like the World Animal Health Organization can also be a key player. The different partnerships that do exist, the industry associations, the IABS, AFSA itself, APAA, so we can definitely take out as a probably next step to come back and maybe have some targeted conversations so that we can be more aligned and understand how we can carry on with the conversation and potentially really create something even more concrete that's just a forum to discuss, but maybe something more practical can be thought. So from my side, thank you, but I let Joris and then David to officially close this workshop. Thank you.
Thank you, Laura. Sorry. Thank you, Laura. Nothing to add to that. You made a perfect resume. So I'd like to thank all of you for your very active participation, and also those who are in hybrid by teleconference. So thank you very much for your participation, and especially those who have a big different time zone. So thanks a lot, and collaboration, expertise, exchange. So these are the keywords, and Mark will make a report, and we hope to publish that as soon as possible. And I think he will circulate that before, as usual. And this can be, this report will be very important, will be the basis for further discussions. My hope is that we can continue this kind of meetings on a global level, and probably more specifically also to Latin America, Africa, and Asia, so that we slowly, step by step, get to this global harmonization. So thanks a lot, and I wish all of you a good trip back. So David John.
Thank you, and there's not a lot left for me to say, and I'm cognizant I'm now the last person standing between you and your flights home. But I think I'd just like to say thank you to everybody online and in the room. I think we've had some very good, very fruitful, and some very open discussions going on. I think coming out of it, I think it's been highlighted, we've got some fruitful avenues to go forward and talk about, maybe some more work on collaboration around training, discussions about new techniques at more basic levels, not just the products. And so I think it's very good to hear that, and I think it bodes well for the future. So I think very good. Thank you, everybody. And also just one last thanks to those sat at the back of the room who've made sure this meeting's kept running smoothly and without interruption. So thank you very much, and goodbye.